Welcome back to the Deep Dive. Today, we're plunging into, well, a frontier that's really shifting, isn't it? From just theory to actual, tangible patient benefit. Cancer vaccines.
It really is. For decades, the idea itself felt almost like science fiction.
Exactly. But now it's fundamentally reshaping how we even think about fighting cancer.
Yeah, absolutely. When most people hear vaccine, the first thought is preventing infectious diseases, polio, measles, more recently, obviously, COVID-19. Huge public health wins.
Monumental wins.
Yeah.
Those vaccines have always been about defense, right?
That's it.
Protecting us from threats before they get a fight.
Precisely. Protecting us from things outside the body.
But what if?
Yeah.
What if that same concept, harnessing our own immune system, could be turned inward against something already established?
And that's the crux of it. What if it could actively treat diseases like cancer? That's where this whole field gets incredibly exciting.
That's exactly what we're exploring today. This deep dive is all about how these, well, groundbreaking therapies are moving beyond just prevention. They're actively training our body's own immune system to fight existing tumors.
It's a strategic shift, really, from pure defense to active offense within the body, empowering our own systems to become the therapy.
So for this journey, we're leaning heavily on a fantastic review. It's from teams of the Tisch Cancer Institute, the Precision Immunology Institute, and the GENOMIC Institute. Really top names in the field.
Published in The Lancet, no less. It's a really comprehensive look at the current trials, where things are headed. It's authored by people cruelly guiding this field, like Oren Pale, Matthew Lin, Theodore Agnosto, Brian Brown, Joshua Brody. These are the experts.
Exactly. And their work gives us this critical lens, doesn't it, to understand not just what's happening, but why some strategies are working, why others are hitting roadblocks.
Yeah, it's more than just data. It's insight into the breakthroughs, the surprising successes, but also the real challenges that are still very much there. Helps cut through the jargon, I think.
Definitely. So our mission for you listening is clear. By the end of this, you'll get the different types of cancer vaccines, how they work with our immune system, the cutting-edge science, the hurdles. We want you to have that aha moment, quickly, thoroughly.
Let's do it.
Okay, so to really get this shift, we need to start with a basic distinction. It's crucial. Vaccines that prevent versus vaccines that actively treat. Prophylactic versus therapeutic.
That's the starting line, yeah. Prophylactic vaccines are the ones we know best, like for hepatitis B or HPV.
Right, preventing liver cancer or cervical cancer by targeting the virus causing it.
Exactly. They stop the cancer before it even starts by stopping the pathogen. And they've been incredibly successful, reducing cancer rates globally. Huge impact.
Huge. But the real game changer for this discussion, our focus, are the therapeutic cancer vaccines. Not prevention, but treatment for existing tumors.
Right, teaching your immune system to go after cancer cells already present in your body.
And the source is clear. These are now showing, and I quote, real promise in clinical settings. This isn't just theory anymore.
And that promise is, starting to show up in results. In the adjuvant setting that's after initial treatment like surgery to stop recurrence, we're seeing good signs in melanoma, definitely.
Okay.
But also, quite surprisingly, in pancreatic cancer. PDAC.
Pancreatic cancer. That's notoriously tough for immunotherapy.
Incredibly tough, low response rates historically. So seeing vaccines potentially clearing out those microscopic leftover cells, preventing relapse in PDAC, that's a massive step.
It really is. And then there's the macrometastatic setting for larger established tumors.
Yeah, even when the cancer has spread, certain types, called in situ vaccines, have induced what they call systemic regression. Meaning you inject a vaccine into one tumor and then you see other tumors, ones you didn't even treat directly, shrinking or disappearing elsewhere in the body.
Wow. That's systemic power from a local treatment.
Exactly. We've seen hints of this in advanced lung cancer, breast cancer, lymphomas. It shows the potential for a truly body-wide immune response triggered by the vaccine.
Which brings us right to the core mechanism, doesn't it? The immune system's hit list. Tumor antigens and T cells.
Precisely. The fundamental idea is that these vaccines work by expanding your T cells. These are the immune system's precision killers.
Okay.
And they expand them specifically against certain molecular flags called antigens found on the tumor cells.
So how do these T cells get, activated and pointed at the right target?
It involves specialized cells called antigen-presenting cells, or APCs. Dendritic cells are the key players here. They're the most potent activators.
Right.
They basically grab those tumor antigens, those unique flags from the cancer, and they display them for the T cells to see.
Like showing them a wanted poster.
Exactly like that. And once a T cell sees its target antigen presented by an activated APC, it just goes wild. It proliferates, multiplies thousands of times, creating this RNA program to hunt down and kill any cell showing that specific antigen.
And the precision is incredible, right? The source mentioned distinguishing proteins that differ by just one amino acid, like KOPS versus KASG12D, which leads to maybe the biggest challenge, finding the right target in the first place. Cancer cells are our cells, just mutated, not like a foreign virus with totally different proteins.
That's the core difficulty, isn't it? Defining the optimal antigen. How do you target the cancer without hitting healthy tissue?
So what kinds of antigens are we actually targeting?
Primarily two types. First, tumor-specific antigens, or neoantigens. These come from mutated proteins, or sometimes viral proteins integrated into the cancer cell DNA. They are genuinely new to the body.
So the immune system has never seen them before on healthy cells.
Exactly, which makes them theoretically the optimal targets. They're unique to the cancer, so attacking them is less likely to cause autoimmune issues, attacking healthy self-tissue. Think of it as a unique barcode on the cancer.
Okay, neoantigens. What's the other type?
Tumor-associated antigens. These are technically normal proteins, but they're expressed at much, much higher levels on cancer cells than on most healthy cells. Or they might be proteins normally restricted to specific tissues, like the testes, but then get turned on in a lung tumor, for example.
The source mentioned NYES1 as an example. Normally in testes, but pops up in other cancers.
That's a classic example. But here's the challenge. Because trace amounts might still be present on some healthy cells, targeting these tumor-associated antigens requires a very careful balancing act.
How so?
You need to generate a strong enough immune response to kill the cancer, which has lots of the antigen, without triggering a devastating attack on healthy cells that might have just a little bit. It's about hitting that sweet spot potent anti-tumor effect while avoiding anti-self-immune responses and autoimmune toxicities. Very tricky.
Incredibly tricky. Okay, so we've got the why the potential and the how the immune mechanism. Now, let's dig into the different strategies scientists are using. How are these vaccines actually classified?
Right. The source gives us a good framework. They're basically classified based on two main things. First, how the antigens are chosen. Are they known beforehand, or does the immune system discover them?
Okay.
And second, where that critical interaction happens between the antigen and the antigen-presenting cells. Does it happen in a lab dish or right there in the body?
Got it. So let's start with the first big category, defined antigen vaccines. The name implies we know the target.
Exactly. These vaccines target antigens that are identified before the vaccine is made. They're either synthesized chemically or encoded in DNA or RNA, which tells our cells how to make the antigen.
Like a pre-programmed missile.
A good analogy. And within this defined group, there are two key types. First, personalized vaccines.
Tailor-made for each patient.
Precisely. It's bespoke medicine. They take a biopsy of the patient's tumor, use advanced omics tech DNA, RNA sequencing often combined with AI, to pinpoint the unique neoantigens specific to that individual's cancer.
So my vaccine would be totally different from yours based on our specific tumor mutations.
Absolutely unique. Then they manufacture a vaccine containing just those personalized targets.
Okay, that's incredibly specific. What's the other type within defined antigens?
Shared, or sometimes called universal vaccines. These are more off the shelf. They target antigens, either known neoantigens or tumor-associated antigens that are commonly found in a large percentage of patients with a specific type of cancer.
Ah, so if lots of people with, say, lung cancer have the same mutation.
Exactly. The source gives KRASG12D as a prime example. It's common in many non-small cell lung cancers and pancreatic cancers. Or BRAV600E in melanoma, about half of patients have that one.
So because these are shared across many patients, you can potentially make one vaccine that works for all of them.
That's the idea. It makes manufacturing and deployment much more scalable than the fully personalized approach.
But the line can be a bit blurry, can it, between shared and personalized?
It can. The source makes a good point. The distinction lies in whether the neoantigen can be detected with standard clinical testing, which is continuously evolving. As tests get better, something that was once rare and personalized might become recognized as a common shared target.
Okay, that makes sense. So if defined vaccines are about knowing the enemy up front, what about the anonymous antigen vaccines? How does that work if the target is, well, anonymous?
It's a clever, different strategy. Instead of identifying the target first, these vaccines aim to make the tumor reveal its own antigens directly to the immune system in place.
So you're not telling the immune system what to look for.
You're creating the conditions for the immune system to find the targets itself. The vaccine components trigger processes within the tumor like cell death and inflammation that cause the tumor cells to release a whole mix of their antigens. The immune system then discovers and responds to whatever it finds immunogenic. The specific targets might remain unknown or anonymous to us, but the immune system figures it out.
That's fascinating. It sounds almost more natural, letting the body do the detective work.
In a way, yes. And it actually has this amazing historical backstory. William Coley, back in the 1890s.
Injecting bacteria into tumors.
Yeah. He injected bacteria, or bacterial toxins, directly into sarcomas and saw some remarkable regressions. It was basically an early crude version of stimulating an immune response at the tumor site to fight the cancer. An early in situ anonymous antigen approach, even if you didn't call it that.
Incredible that the concept goes back that far. So what are the advantages today of this let the immune system decide approach?
A key potential advantage is breadth. It might expose the immune system to a wider range of antigens than even sophisticated AI can predict for defined vaccines. Think complex things like fusion proteins, weird splice variants, bits of endogenous retroviruses, or proteins modified after translation things often missed by standard prediction algorithms.
So it casts a potentially wider net.
It could, yeah. However, there's a critical requirement. You absolutely need efficient uptake of these released antigens by APCs, and those APCs need to be strongly activated. The best antigens in the world won't help if they aren't presented properly to the T cells.
Activation is key.
Yeah.
And like the defined vaccines, these anonymous ones are also classified by where that antigen APC meeting happens.
Yes. Two main ways. Ex vivo, meaning outside the body. Here, you take tumor material, whole cells or lysates, from the patient and essentially mix it or fuse it with the patient's own APCs in the lab.
So the training happens in a dish.
Right. Then those educated APCs are infused back into the patient. It's personalized, but the immune interaction happens ex vivo.
And the alternative?
In situ, meaning in place. Here, the vaccine components, which are often off the shelf, are injected directly into the tumor. The tumor cells die, release antigens, and the patient's own APCs get activated right there at the site. The personalization happens in vivo within the patient's body, much less complex logistically than the ex vivo method.
Okay, really clear breakdown. Defined versus anonymous, and then ex vivo versus in situ for the anonymous ones. Let's dive into the clinical side now, starting with those defined antigen vaccines. Specifically, the personalized neoantigen ones, the bespoke approach.
Right. Quick recap. Biopsy, identify unique neoantigens using genomics AI, manufacture a patient-specific protein RNA DNA encoding those targets, add immune activators, inject.
And the early attempts, like for lymphoma, targeting idiotypes, they didn't quite pan out in phase 3. What went wrong?
It seems the early formulations might have been suboptimal. Maybe not enough antigen or the wrong length peptides, or crucially, not enough effective mobilization and activation of those critical APCs. It really drove home how complex it is. You need the right target and the right presentation and the right activation. Get any part wrong and it fizzles.
Important lessons learned, but then came a big one. Melanoma, mRNA 4157, Keynote 942. That seemed like a real turning point.
A huge turning point, yes. This was a phase 2 trial, 157 patients, high-risk melanoma after surgery. They got the personalized mRNA vaccine plus pembrolizumab, an anti-PD-1 drug.
And the results?
Really remarkable. At 18 months, relapse-free survival was 79% for the vaccine group versus 62% for pembro alone. Distant metastasis-free survival, 92% versus 77%. Statistically significant improvements, a major signal.
Those are big numbers. How does that mRNA vaccine work technically? What's special about it?
It's mRNA encoding up to 34 patient-specific neoantigens, but the key is it's encapsulated in the same lipid nanoparticle LNP technology used in, for example, Moderna's COVID vaccine.
Reusing that successful tech.
Exactly. And crucially, those LNPs aren't just delivery vehicles. They actually have adjuvant properties themselves. They help activate dendritic cells, so you get the personalized antigen payload delivered effectively and an immune boost from the packaging itself. Very clever synergy.
Very clever. But the source mentioned some caveats with that phase 2 study. Limitations.
Yeah, transparency is important. It used a one-sided statistical analysis. There were some imbalances between the groups in things like PD-L1 levels and tumor mutational burden. They didn't have a control group getting just the empty LNP or a non-specific mRNA, which would help isolate the vaccine effect.
Okay.
And randomization wasn't fully completed due to the pandemic, affecting follow-up a bit. But these are things typical of phase 2. And the good news is a large phase 3 trial is ongoing, NCT 059333577, finishing around 2029, which should give much more definitive answers.
And the standard of care keeps changing too, right?
Yeah.
Things like neoadjuvant immunotherapy before surgery.
Absolutely, that could change where adjuvant vaccines fit in. But the source suggests there's still likely a strong role for vaccines like this after surgery as an adjuvant to really consolidate the response and prevent relapse, even alongside newer standards of care.
Okay, now for something really unexpected, pancreatic cancer, autogene cevumeran. You mentioned PDHE is tough, low TMB usually.
Yeah.
What was so surprising here?
This was a small study, non-randomized, so we need caution. But in patients with resectable PDA, about half, 50%, showed T-cell responses to at least one neoantigen in the vaccine. And importantly, that immune response correlated with longer recurrence-free survival.
That's significant for PDA.
It's huge. It suggests that even in these traditionally cold, immune-resistant tumors, if you can find the right neoantigens and present them effectively, you can generate a meaningful, clinically relevant immune response. It challenges assumptions.
So what's next for that one?
Given the promise, it's now in a randomized phase 2 trial, NCT 059683326. That will be critical to see if these early findings hold up in a larger, more rigorous setting.
Are there other personalized approaches showing promise? Different technologies, maybe?
Definitely. NEO-PVA01, another peptide-based neoantigen vaccine used with an adjuvant, showed T-cell and clinical responses in melanoma, lung, and bladder cancers. And there's ChAd68, that's a chimpanzee adenovirus vector carrying self-amplifying mRNA.
Self-amplifying.
Yeah, the idea is to get more sustained antigen expression compared to standard mRNA, which can be transient. Early results in advanced solid tumors showed it induced long-lasting T-cell responses, which is exactly what you want for durable anti-tumor immunity.
So clearly moving forward. But what are the big picture challenges still facing these personalized vaccines? Why aren't they everywhere yet?
The potential is huge, but the practical hurdles are real. The biggest ones are the lead time it takes weeks or months to analyze the tumor, design, and manufacture a personalized vaccine, and the cost and resource intensity. It's complex, bespoke manufacturing for every single patient.
Right.
Plus, current algorithms for picking neoantigens aren't perfect. They might miss important types, like antigens modified after the gene is transcribed into protein, because they mostly rely on DNA sequencing.
But progress is being made on those fronts.
Absolutely. Computational methods are getting smarter at predicting which mutations will actually be immunogenic, and manufacturing processes are being streamlined to cut down production time and cost. These are solvable problems, but they take time and innovation.
Okay, let's switch to the other defined antigen category, the defined shared antigen vaccines, the off-the-shelf potential, aiming to overcome those personalization bottlenecks.
That's the core rationale, yes. Target common vulnerabilities shared across many patients, makes development and deployment potentially much simpler and cheaper.
And the very first FDA-approved cancer vaccine, Sipuleucel T, falls into this category, targeting a shared antigen.
It does, approved back in 2010 for metastatic prostate cancer, a real landmark.
How did it work again? It was autologous, right? Using the patient's cells.
Yes. They take the patient's monocytes, culture them ex vivo with PAP prostatic acid phosphatase, a common prostate cancer antigen fused to GM-CSF, an immune stimulator, then infuse those educated cells back into the patient.
Training the patient's own cells against PAP. What was the clinical impact?
It showed a statistically significant overall survival benefit, about a 4-month improvement in median survival compared to placebo, 25 months versus 22 months. A clear, measurable benefit.
A clear benefit.
Yeah.
Yet the source says its use has been modest. Why? What's the cautionary lesson here?
This is really important. Despite the survival gain, it didn't often cause clear tumor shrinkage. Objective responses were rare. It was logistically complex, requiring specialized centers for the cell processing.
Okay.
And crucially, other, easier-to-administer systemic therapies for prostate cancer emerged around the same time or soon after, offering similar or better efficacy. So while it proved the concept, practical challenges and the evolving treatment landscape limited its uptake. It shows that survival benefit alone isn't always enough if it's complex and other options are better or simpler.
A really key lesson. Efficacy, logistics, competition, it all matters. What about the melanoma GP100 vaccine? Another shared antigen approach.
Yeah, GP100 is a tumor-associated antigen in melanoma. Early trials combining it with high-dose IL-2 looked promising.
And the phase 3.
It also showed a survival benefit, 18 months versus 11 months for IL-2 alone. Good on paper.
But there's always a but, isn't there?
The but was toxicity. High-dose IL-2 is notoriously harsh.
Yeah.
Considerable toxicity, including treatment-related deaths. It's a huge barrier. And then a later trial combining GP100 with ipilimumab, another immunotherapy, showed no added benefit from the vaccine. So severe side effects and lack of synergy limited its use.
Again, highlighting that the therapeutic window benefit versus harm is crucial. And this brings up that issue of targeting just one antigen. Tumor heterogeneity means the cancer can just evolve around it, right? Antigen escape.
Exactly. That's a major reason single antigen approaches often fail long-term. Tumors are diverse internally. Kill off the cells with antigen X, and the cells without antigen X just take over. That's why multi-antigen vaccines are a big focus now.
Makes sense. Attack on multiple fronts. What are some examples using shared antigens?
OSC 2101 is one. It's a peptide vaccine hitting 5 different tumor-associated antigens common in non-small cell lung cancer, P53, HER2, CEA, MAGEs. In a specific subgroup of patients who'd become resistant to checkpoint inhibitors, it showed a survival benefit 11 months versus 7.5.
Promising, but needs confirmation.
Yes, the study was stopped early due to COVID, so it was underpowered. Results need verification in a larger trial. Another is FixVac, a liposomal RNA vaccine targeting 4 melanoma antigens. Showed responses even in patients who'd already had checkpoint inhibitors, which is encouraging.
So hitting multiple targets seems key. Are we also getting better at selecting the best shared antigens and presenting them more effectively?
Definitely. On selection, techniques like mass spectrometry are being used to directly identify which proteins or peptides are actually presented on tumor cells. IMA970A, a 16-peptide vaccine for liver cancer, was developed this way and induced T-cell responses that correlated with better survival, better selection.
And better presentation, getting them to the immune system effectively.
Yeah, novel delivery strategies, like lymph node targeting. ELA-002 uses special molecules to chaperone the vaccine antigens, in this case mutant KRAS peptides, directly to the lymph nodes, where the immune action really happens.
Taking it straight to headquarters.
Pretty much. And in patients with colorectal or pancreatic cancer with minimal residual disease, it generated strong anti-KRAS T-cell responses and even cleared that minimal disease in many patients. Very clever delivery.
Very clever. What about tackling the tumor's defenses directly? That immunosuppressive microenvironment.
That's another exciting angle. Vaccines like IO102, IO103 target the suppressors themselves, IDO and PD-L1.
Vaccinating against the tumor shield.
Exactly. Disarm the defenses. In melanoma, combining these with anti-PD-1 led to really high response rates, 80% overall response, 50% complete remission, and T-cell responses in almost everyone. It suggests that tackling suppression directly can make other immunotherapies work much better, turns cold tumors hot.
Incredible potential there. And finally, for defined vaccines, this idea of epitope spreading. Can you explain that again? Why is it so important?
Epitope spreading is like a positive feedback loop. The vaccine primes T cells against the specific antigens in the vaccine, epitope A, B, C. Those T cells attack the tumor, kill cells, and cause the release of other tumor antigens, epitope D, E, E, F, that weren't in the original vaccine.
Okay.
APCs then pick up these new antigens and present them, broadening the T-cell response to target D, E, F as well. The immune attack gets wider and deeper over time.
So the immune system learns on the job, expanding its target list.
Precisely. The NeoVax trial showed this T-cells responding to antigens not included in the vaccine. It makes the response more robust and much harder for the tumor to escape just by losing 1 or 2 initial targets. It's a really desirable outcome.
Fantastic. Okay, let's pivot now to the anonymous antigen vaccines, starting with the ex vivo type where the training happens in the lab.
Right. Recap. Take patient's tumor material, cells or lysate, mix-fuse it with their own APCs in a dish, let the APCs learn the antigens, then infuse those APCs back into the patient.
And the example here was DCVax-L for glioblastoma, that aggressive brain cancer. Phase 3 results showed longer survival, but were contentious. Why the controversy?
Well, the survival difference was there about 3 months median improvement compared to external controls. But interpreting it was tricky. Glioblastoma often causes brain swelling after treatment that can look like tumor growth on scans pseudoprogression. That complicates measuring how well the treatment is working initially. Also, many patients in the original control group eventually received the vaccine, crossover, making a direct randomized comparison impossible. They had to use historical or matched external controls, which is always less ideal, and opens the door to debate about comparability. So a positive signal, but complex trial issues.
Understandable. What about Gemogenobatocell T for ovarian cancer? How did that one work?
That's another autologous ex vivo approach. They take the patient's tumor cells, transfect them with GM-CSF to attract APCs, and also with an RNA molecule to shut down immunosuppressive factors like furin and TGF-beta that the tumor makes.
So making the tumor cells both more visible and less suppressive before putting them back. Did it work?
The Phase 2b trial showed improved recurrence-free survival overall, 11.5 versus 8.4 months. It wasn't quite statistically significant for everyone, but a specific subgroup patients without BRCA mutations had a significant benefit in both recurrence-free and overall survival.
Interesting. So maybe it works particularly well in certain patient subsets.
That's what it suggests, and that finding has prompted plans for a follow-up trial focusing perhaps on that BRCA negative group. It highlights the importance of identifying biomarkers to predict who will benefit most.
Makes sense. And there were also dendritic cell fusion vaccines for leukemia and myeloma, fusing tumor cells directly with DCs.
Yes, the idea is maximum antigen presentation by literally fusing the tumor cell with all its antigens to the best APC, the dendritic cell. Early studies looked promising induced anti-tumor T cells, some clinical responses.
But mixed results in larger trials.
Yeah. A big Phase 2 in multiple myeloma showed it boosted T cells but didn't translate into significant clinical benefit. Disappointing. However, a Phase 1 in acute myeloid leukemia looked much better, with many patients staying disease-free long-term. So again, efficacy seems quite dependent on the specific cancer type.
What's the overall verdict on these ex vivo anonymous approaches then?
The source sums it up as having shown some proof of principle but have yet to prove clinical benefit consistently in large trials. The big limitations are still the resource intensity of production, its complex, individualized lab work, and the difficulty in precisely measuring the T-cell response when you don't know the exact antigens being targeted. That makes optimizing the vaccines and designing big trials harder.
Okay, let's move to the other anonymous approach, in situ, the tumor as the vaccine factory. Conceptually simpler, you said.
Conceptually, yeah. Inject off-the-shelf components directly into the tumor. Let the body do the antigen release and APC activation locally, avoids all that ex vivo cell handling. But, as the source notes, while simple in concept, there's still lots of room for improved understanding to optimize design. Getting it just right in vivo is complex too.
What are the absolutely essential components you need for an in situ vaccine?
Two key things. One, something to cause immunogenic cancer cell death. You need to kill the tumor cells in a way that releases their antigens effectively and sends danger signals to alert the immune system.
Not just kill them quietly.
Right. And two, an inducer of inflammation or an adjuvant, something to really activate those APCs that arrive on the scene, especially dendritic cells, so they efficiently present the released antigens to T cells.
Kill loudly and activate the responders. And you mentioned type 1 dendritic cell. DC1s are particularly important here.
Absolutely critical. DC1s are uniquely good at presenting antigens to CD8 T cells, the main cytotoxic killers we need. But DC1s are rare, often inactive, and tumors try to keep them out. So a major focus of modern in situ vaccines is specifically mobilizing and activating these elite DC1s right within the tumor.
Targeting the best activators. Now, oncolytic viruses fit here, right?
Yeah.
Like TVC.
Yes, TBE is a perfect example. It's an engineered herpes virus that infects and kills tumor cells, immunogenic cell death, and is also engineered to produce GM-CSF, an APC activator, though maybe not the best one for DC1s.
So it does both jobs. The Phase 3 in melanoma showed benefit?
It did. Better complete response rate, 17% yes is 1%, and better overall survival, 23 versus 19 months, compared to GM-CSF alone. FDA approved.
But its use has been only moderate. Why?
Partly timing it came out just as highly effective checkpoint inhibitors were becoming widespread. Combinations with those checkpoint inhibitors showed only modest TVEC benefit. And the thinking now is that GM-CSF might be suboptimal in mobilizing type 1 dendritic cells compared to newer agents. The field moved fast.
It's always evolving. Are there other virus-based approaches that maybe activate DCs better?
Yes, nadofaragine ferdinebec. It's not an oncolytic virus, doesn't replicate, but it's an adenovirus vector carrying the gene for IFNA2B, a very potent DC activator. In bladder cancer patients unresponsive to standard BCG therapy, it achieved complete responses in over half of them, 53.4%. Huge result. Shows the power of potent DC activation.
That's impressive. And you mentioned FLT3L earlier as being even better for DC1s. especially with radiation.
Right. FLT3L seems to be a more specific and potent mobilizer of type 1 dendritic cells than GM-CSF. And radiation is a great way to induce that immunogenic cell death locally. So the combination looks really powerful.
What are the clinical results showing for that combo?
Very promising. In non-small cell lung cancer, FLT3L plus localized radiation led to shrinkage of tumors outside the radiation field at abscopal effect in about a third of patients, systemic immunity from local treatment. In lymphoma, combining FLT3L, radiation, and poly-ICLC, a TLR3 agonist that strongly activates DC1s, led to T cells trafficking to distant sites, and some patients had deep, durable remissions lasting years.
Even in tough refractory lymphoma.
Yes. And then the really exciting step, adding anti-PD-1 to that FT3L radiation poly-ICLC combo.
What happened then?
Preliminary results showed remissions not just in refractory lymphoma, but also in certain types of breast cancer, where anti-PD-1 alone usually doesn't work well. This offers strong hope for using this kind of in situ vaccine to make cold tumors responsive to checkpoint inhibitors, a major potential expansion.
That really could open up immunotherapy to many more patients. Are there other immune stimulants used in situ besides FC3L and poly-ICLC?
Oh, yes. TLR agonists in general have been tried a lot, like tilsatolimod, TLR9, but enthusiasm for them as single agents has cooled a bit after early promise didn't always hold up in bigger trials. IL-12 is another potent one, a T-cell activator.
How is IL-12 done?
Intratumoral IL-12 electroporation plus anti-PD-1 showed high complete remission rates in melanoma, 36%. The big challenge with IL-12 is managing toxicity if it gets systemic. It's powerful, maybe too powerful if it's not kept localized. Really highlights the need to keep these potent immune stimulants precisely localized to the tumor.
Tricky balance. So, overall assessment of in situ vaccines. Huge potential, but hurdles remain.
Definitely huge potential. They've shown they can cause systemic tumor regressions in patients with advanced stage, high tumor burden, and chemotherapy-resistant cancers. That's a major achievement. The key limitations right now are the need for direct intratumoral injection for many components, limiting access, and still needing to overcome that suppressive tumor microenvironment fully.
With solvable challenges.
The source thinks so. It points towards innovations like systemically administered agents that target DC1s, like CD40 bispecific antibodies. If you can deliver the activation systemically but have it act primarily at the tumor, that bypasses the need for direct injection and could be a game changer for accessibility.
Okay. Incredible progress and potential. Let's step back now and look at the bigger picture. The overall challenges and future directions for the whole field of cancer vaccines. First off, which tumors are the best candidates for which type of vaccine?
It's not one size fits all. Generally, personalized vaccines targeting neoantigens are most feasible for tumors with high tumor mutational burden, high TMB.
Right.
Like melanoma, lung cancer, bladder cancer. More mutations usually mean more neoantigens to target.
Makes sense.
In situ vaccines currently are more practical for tumors you can actually inject into and that respond reasonably well to things like radiation. So accessible breast cancers, lymphomas, some lung cancers.
But you mentioned that accessibility issue might change.
Exactly. If we can develop systemically delivered in situ activators that target tumor-specific surface markers like mesothelin, PSMA, HER2, then the need for easily injectable tumors might become unnecessary. That would vastly broaden the scope.
And the rules aren't absolute, are they? Pancreatic cancer, low TMB, but still responding.
That's the crucial point. PDAC's response to vaccines, despite low TMB and checkpoint inhibitor resistance, shows that tumors that do not respond to checkpoint blockade might still have targetable antigens and benefit from vaccines. It opens up possibilities beyond the usual hot tumor suspects.
So assuming you can target a tumor, what makes an optimal antigen? Quantity, quality.
Both seem important. More antigen targets, higher total antigen content seem better, likely reducing escape. And targeting driver mutations, the ones essential for the cancer's growth, should be better than targeting random passenger mutations because the cancer can't easily afford to lose them.
Hit them where it hurts, functionally. And this is where anonymous vaccines have that potential edge again.
Potentially, yes. By letting the immune system identify the antigen, they might pick up on important targets missed by prediction algorithms, like those post-transcriptionally modified proteins or fusion products. They leverage the immune system's own pattern recognition.
And specificity still matters. Neoantigens over tumor-associated antigens.
Generally preferred, yes. Targeting truly tumor-specific neoantigens minimizes the risk of autoimmunity attacking healthy tissues. It's safer and potentially more effective as you don't have to overcome self-tolerance.
What about peptide length? You mentioned the long peptides are better.
Yes, the consensus is that longer peptides, maybe 15-30 amino acids, are generally better than short ones, 8-10 amino acids, and both are better than whole proteins.
Why is that?
Longer peptides can be processed and presented by APCs on both MHC class I to activate CD8 killer T cells and MHC class II to activate CD4 helper T cells. Getting both arms of the T cell response working together leads to a much more robust and sustained anti-tumor effect. Short peptides mainly hit MHC ICD8.
Coordinated attack is better. And AI is helping sort through all this antigen complexity.
Hugely important. Machine learning is being trained on vast data sets, pathogen epitopes, preclinical vaccine results, clinical trial data to get much better at predicting which specific mutations, in the context of a patient's specific immune system genetics, HLA type, are most likely to be immunogenic. Better antigen prediction tailored to each patient.
And the ultimate dream is using this predictive power for prophylactic vaccines, preventing cancer in high-risk people.
That's the long-term vision. Imagine identifying shared high-risk mutations, like in Lynch syndrome patients, and developing a vaccine to prime the immune system before cancer even develops. A huge paradigm shift from treatment to prevention based on genetics.
Mind-blowing potential. Okay, beyond the antigen itself, what other vaccine properties really matter for efficacy? Activation, delivery?
Absolutely crucial. You can have the perfect antigen, but if it's not presented well, it won't work. You need enough APCs, they need to take up the antigen efficiently, and they need to be properly activated.
And we're getting better at activating them. FLT3L seems better than GM-CSF.
The data points that way, yes. Superior type 1 dendritic cell expansion and T-cell priming with FT3L. Getting those elite DC1s working optimally seems key.
What about adjuvants, the activation signals? Do we know which are best?
We're still figuring that out. The most effective APC activators, adjuvants, or combination thereof is unknown, but studies comparing different ones like poly-ICLC showing benefit over others in glioma are guiding the way. Combinations might be needed.
And the delivery format. RNA versus DNA versus peptide, intramuscular versus intravenous.
All of that matters. The molecular format, the delivery type like LNPs, the route of administration, they all influence the type, strength, and location of the immune response. We need more direct head-to-head comparisons in patients to really nail down the optimal delivery for different situations.
So many variables. Which brings us to trial design. How do we best test these vaccines? Adjuvant versus metastatic setting.
They have different goals. Adjuvant trials, after surgery, minimal disease, aim to increase cure rates by preventing relapse. Metastatic trials, established widespread disease, aim for survival prolongation and systemic tumor regressions.
And different vaccine types have shown strengths in different settings so far.
Generally, yes. Anonymous in situ vaccines have shown more precedent in shrinking established macrometastatic disease. Defined, personalized neoantigen vaccines have shown more success so far in the adjuvant setting, clearing minimal residual disease. But those lines might blur as the technologies improve.
What's the biggest roadblock in interpreting trial results right now? Measuring the immune response.
That's a crucial challenge. We need reliable immune response measurements that predict clinical responses. For COVID vaccines, antibody levels work well as a surrogate. For cancer vaccines, it's mostly T cells, and those are hard to measure reliably and consistently in a way that predicts if the patient will actually benefit long term.
We need better biomarkers for T cell responses.
Exactly. Without good surrogates, we have to wait longer for clinical outcomes, which slows down development. The good news is, better assays are being developed looking at T cell function, tracking specific T cell receptors. Getting those validated will be huge for accelerating progress.
What an incredible journey we've taken through this field today, from the basic concepts to the really cutting-edge trials and future hopes.
It's truly remarkable. I mean, the success of other immunotherapies, checkpoint inhibitors, CRRTE, proved the principle. Our immune system can beat cancer, sometimes durably. Cancer vaccines are now emerging as this distinct, powerful tool within that arsenal, specifically focused on training and directing that immune power.
Yeah, enhancing the strength, the breadth of the T-cell response. We're seeing it prolong remissions, induce those systemic regressions, even in tough metastatic cancers. It's genuinely transformative.
It is. But, as we've covered, the challenges are still significant. We need those better immune surrogates. We need to boost efficacy across more cancer types, more patients, and critically, figure out how to do it all practically and affordably on a global scale.
Looking out over, say, the next decade, what are the biggest changes you expect to see? Where's the momentum heading?
For in situ vaccines, The big leap has to be tumor-targeted systemic delivery. Moving beyond needing accessible tumors for injection will open them up to far more patients. That's key.
Right.
For defined vaccines, especially personalized ones, it's all about large randomized trials to prove efficacy definitively and driving down manufacturing costs and complexity to make them accessible. Scale and cost reduction.
It feels like we're truly at an inflection point. Decades of development,
Yeah.
and now we're seeing real, tangible patient benefits. The momentum feels different now.
It absolutely does. We're
Yeah.
gaining such a deeper understanding of the biology, and the tools are getting so much better, what the source calls moving towards total programmable control of the anti-tumor immune response. Optimizing DC1 mobilization, activation, antigen loading, getting all those pieces right, promises a much more powerful, reliable vaccinal effect, potentially systemically.
Total programmable control of our immune response against cancer. That's profound. So a final thought for you listening. As we gain this level of precise control, what new breakthroughs do you think are just around the corner? And how might this fundamentally change what we even mean by a cure for cancer? Something to ponder as this exciting field continues to race forward.